Good morning, ma'am. Good morning, classmates. Maayong buntag sa tanan. We are here to present to you our presentation about the citric acid or tricarboxylic acid cycle, or also known as the Krebs cycle. The objectives in this cycle are: number 1, to deeply understand what really a Krebs cycle is. 2, to determine who discovered the cycle and what is the meaning of the names behind it. 3, list down the steps of the cycle. And 4, to find out the summary reaction of it. But before we start, here are the reporters in this presentation. We have Julmaylin Tugas, Alexis Nicole Nieva, and Sami Yudelia. So, what is a Krebs cycle? The Krebs cycle, also known as the citric acid or tricarboxylic acid cycle, is a series of reactions that produce molecules that can be used as building blocks for a variety of important processes, including the synthesis of fatty acids, steroids, cholesterol, amino acids for protein building, and purines and pyrimidines for DNA synthesis. It is one of the major steps or processes of cellular respiration. It is also the metabolic pathway that connects carbohydrate, fat, and protein metabolisms that later on turn into the central pathway which most of the molecules are being fed. It consists a number of reactions that generate NADH and FADH2, which are in turn to generate adenosine triphosphates. Perhaps most of you are wondering, what are the purpose of the Krebs cycle? The main purpose of the Krebs cycle is to produce energy in our body by connecting the compounds to produce different molecules. It also stored and transported as ATP or GTP. It provides raw materials such as amino acids, NADH or nicotinamide adenine dinucleotide, and ETC or electron transport chain to any parts of the body. So our fact 1, this cycle was named after Hans Adolf Krebs, who discovered it in 1937, and it received a Nobel Prize for Physiology and Medicine in 1953. Fact 2. The other names for the Krebs cycle are citric acid cycle because citric acid is both the first product and the final reactant, and it is also known as the tricarboxylic acid cycle because of the involvement of the 3 carboxyl groups. But before we proceed, let's define first, what is carboxyl group? It is defined as having a carbonyl and hydroxyl group, both linked to a carbon atom. To refresh your memory, a carbonyl group is a carbon double bonded to an oxygen, and a hydroxyl group is an OH group or oxygen-hydrogen group. Fact 3. It takes place in the mitochondria of eukaryotes. It is an important aspect of the production of adenosine triphosphate or ATP, and it also takes place in the cytosol in the prokaryotes. Lastly, this cycle was one of the earliest established components of cellular metabolism and may have originated abiogenic. Abiogenesis. It means that life originated from non-living things. What do we mean by central metabolic pathway? When we eat food like fish for protein, bread for carbohydrates, then fats, it is digested and absorbed and is first processed by initial metabolic pathways. These initial metabolic pathways generate some common products that are used by the central pathway like the Krebs cycle to produce energy. Like if we take the example of carbohydrates or our bread, Carbohydrates are digested into simple sugar like glucose, and the glucose is first processed by initial metabolic pathways like glycolysis. Glycolysis produces pyruvate, and similar products are produced by the initial metabolism of fats and proteins of fats and proteins as well. The pyruvate is then fed into the central pathway, that is the Krebs cycle. So the Krebs cycle provides a central pathway that connects carbohydrates, fats, and protein metabolisms. How does the Krebs cycle produce energy? So let's take the example of metabolism of carbohydrates again. We get simple sugars at the end of digestion like glucose. And after this, glucose undergoes through all the metabolic pathways like glycolysis, Krebs cycle, and electron transport chain. One molecule of glucose produces approximately 36 adenosine triphosphates. But where does this energy come from? The answer to this question is through the process of oxidation. Oxidation is defined as a process in which an electron is removed from a molecule during a chemical reaction. In the oxidation reactions of the Krebs cycle, the hydrogen atoms or the electrons derived from them do not react directly with oxygen, but pass through a series of hydrogen or electron carriers. And now, see that through glycolysis and additional reaction, we break down glucose into small carbon fragments, which are known as acetate. This acetate is then fed into the Krebs cycle. If you look at the Krebs cycle, it consists of these 10 intermediate molecules that start and end with citrate. When we add acetate to this cycle, a 2-carbon compound, this cycle takes a full turn and releases 2 molecules of carbon dioxide as a waste product, meaning the 2 carbons that we added in the form of acetate are released in the form of carbon dioxide. This carbon dioxide is produced through the process of oxidation that occurs in the Krebs cycle as a result of various enzymes. Electrons are added to NAD to make NADH and FAD to make FADH2. These electrons are subsequently used in the electron transport chain to make ATP through ionic gradients.
Here's a background about the preceding steps of the Krebs cycle. The Krebs cycle starts with this molecule, acetyl-CoA. But where does this come from and what is the role of this in the cycle? This is very important to understand. One of the most important biological pathways that run before Krebs cycle is glycolysis. In this pathway, we break down glucose, which is a 6-carbon compound, into 2 molecules, pyruvate, which is a 3-carbon compound. What happens next is this pyruvate is acted upon by this set of enzymes collectively known as a pyruvate dehydrogenase complex. In this reaction, coenzyme A, which is a coenzyme that also derived from vitamin B5. In this reaction, 2 carbons are transferred from the pyruvate to CoA from the acetyl-CoA. One additional carbon in the form of pyruvate is lost in the form of carbon dioxide. In this reaction, 1 molecule of NADH is also produced. So, this is how acetyl-CoA is produced from pyruvate. This acetyl-CoA is a 2-carbon compound and is the starting point of the Krebs cycle. In this molecule, the coenzyme A acts like a carrier which transfers the 2 carbon attached to it to the Krebs cycle. The first step of the cycle is the combination of the acetyl-CoA with the oxaloacetate. Like I've said earlier, acetyl is a 2-carbon compound and this combined with the oxaloacetate, which has 4-carbon compound. This combination led to the formation of citrate, which is a 6-carbon compound, and this reaction is made possible by the enzyme citrate synthase. Then the citrate undergoes a series of enzyme catalyzed reactions in which it loses the 2 carbon of acetyl-CoA in the form of 2 molecules of the carbon dioxide. The conversions, which involve up to 10 chemical reactions, are all brought by enzymes which use energy generated through oxidation to attach electrons to NAD and FAD. Now, let's look at what happens to citrate. In the next two reactions, the citrate is converted into its isomer, which known as isocitrate. Both these reactions, which I have shown with just one arrow, are catalyzed by the enzyme aconitase. Now, this is an iron-containing enzyme which reorganizes the atoms of citrate to form an isocitrate. You may ask, what was the need to convert citrate to isocitrate? Why can't the citrate just move forward by the pathway? Well, the answer to that is the arrangement of atoms in the isocitrate is better suited for the next few oxidation-reduction reactions that are about to come to Krebs cycle. In the next step, the isocitrate is converted into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase. Now, we come to the first oxidation-reduction reaction in the citric acid cycle. This oxidation-reduction reaction are the one that produce energy to the cycle. And all of these four reactions uses enzyme in them which known as dehydrogenase. But what does this do and how do they produce energy? If you look at the term itself, you can see that it is a removal of hydrogen. Basically, it is enzymes that remove hydrogen from the molecule. Now, as you remember, the removal of hydrogen is also known as oxidation, and the hydrogen that is removed from one molecule must be passed on to some other molecule, and the other molecule that receives the hydrogen gets reduced in return. So this is the basic mechanism of the action of dehydrogenase. They remove hydrogen group from one molecule, oxidizing it, and in return attaching that hydrogen group to some molecule, leading to its reduction. This is why the reactions carried out by dehydrogenase are known as oxidation-reduction reaction. Coming back, we now reach the point where isocitrate is acted upon by the enzyme isocitrate dehydrogenase. This is a two-step reaction, which is the enzyme first removes hydrogen, thus oxidizing it. The hydrogen that is removed is attached to NAD to form NADH. And what happened next is catalyzed by the same enzyme. In this step, a carbon is also removed from isocitrate in the form of molecule of carbon dioxide. These lead to the formation of 5-carbon compound known as alpha-ketoglutarate. So, if you look at the whole process that happened here, this is known as oxidative and decarboxylation. Oxidative because a hydrogen is removed, and decarboxylation because a carbon is also removed. The conversion of alpha citrate to alpha ketoglutarate is followed by the formation of succinyl-CoA. This reaction uses the enzyme alpha ketoglutarate dehydrogenase that catalyzes a similar decarboxylation of alpha ketoglutarate. In this reaction, a hydrogen is also removed to form NADH, and as this is also a type of decarboxylation, a carbon is also removed to alpha ketoglutarate to be lost in the form of carbon dioxide. Also, coenzyme A is to be used in this reaction. Therefore, the product will be the combination that is known as succinyl-CoA. In the next step of the Krebs cycle is the succinyl-CoA converted into succinate. This reaction is catalyzed by the enzyme succinyl-CoA synthetase. The number of carbons remain the same in this reaction. Both of them have 4 carbon each. So, what changes? Succinyl-CoA is a high-energy compound, and the energy in this compound is used to attach an inorganic phosphate molecule to GTP. And GTP gets converted to GTP, which is like a close cousin of ATP, and then later on used to the formation of ATP. The coenzyme A is attached to the succinyl-CoA, also gets released in this reaction. So, what happens here essentially is that the succinyl-CoA loses its all high energy, and its coenzyme A is to be converted to succinate and a molecule of GTP. This is a substrate level phosphorylation, which is a process of producing energy that is discussed in the glycolysis topic. Now we have succinate, a 4-carbon compound that is acted upon by the enzyme succinate dehydrogenase, which is like an iron sulfur protein. So this enzyme removes hydrogen from succinate and it is accepted by FAD to form FADH, and the succinate is converted to fumarate. FAD is the acceptor of hydrogen in this reaction and not NAD because NAD requires more energy than this reaction can provide. The next step of the Krebs cycle is a hydration reaction, which means we add a molecule of water to fumarate to form malate. This reaction is catalyzed by the enzyme fumarase. The last step that happens in the Krebs cycle is oxidation, in which we oxidize malate to oxaloacetate, and the enzyme involved here is the malate dehydrogenase. The NAD gets reduced to NADH, which we will use in the electron transport chain. The oxaloacetate combined with the molecule of acetyl-CoA and leads to the formation of citrate. With the help of enzyme citrate synthase, a water molecule is also added to this form of reaction, but this completes a full turn of the cycle, and this cycle repeats again and again. And here are the summarized 8 written steps that you can review for this topic. Yet, there is still a bit more that we need to know about this cycle. First is the summarized written steps and the energetics of the Krebs cycle, meaning how many ATPs are produced for every 1 molecule of glucose that enters this cycle.
So first coming to the summary reaction of the glycolysis, now the reaction of citric acid cycle is 1 molecule of acetyl-CoA plus 3 molecules of NAD plus 1 molecule of FAD plus 1 molecule of GTP plus 1 molecule of inorganic phosphate plus 2 molecules of water, combined to get 2 molecules of carbon dioxide and 3 molecules of NADH, plus 1 molecule of FADH, plus 1 molecule of GTP, and 1 molecule of coenzyme A. The left-hand side of this reaction are the raw materials that are being used here, and on the right-hand side are the products that are formed in the Krebs cycle. Now in every single turn of the Krebs cycle, we use 1 molecule of acetyl-CoA that combines here with the oxaloacetate to form citrate. 3 molecules of NAD are used in the Krebs cycle that get reduced to 3 molecules of NADH. Here you can see where the 3 NADH molecules are produced in the Krebs cycle. 1 FAD molecule is used which gets reduced to 1 molecule of FADH. This FADH reproduced in the converted of succinate to fumarate. 2 molecules of carbon dioxide are produced for every 1 molecule of acetyl-CoA that enters the Krebs cycle. 1 coenzyme A is produced in the converted of succinyl-CoA, where succinate uses the coenzyme A and 1 molecule of GTP, combines with 1 molecule of inorganic phosphate and leads to the formation of 1 molecule of GTP. 2 molecules of water are consumed, 1 in the synthesis of citrate and 1 here in the hydration of fumarate. That's all for the summary reaction of the Krebs cycle. Now let's talk a little bit about how much energy is produced in the Krebs cycle. Most of the energy that is produced in the Krebs cycle is via the reducing equivalents. The NADH and FADH are subsequently utilized in the electron transport chain. If you look at the placement of these 2 biochemical pathways in the mitochondria, they lie very close to each other in the Krebs cycle, operating in the matrix of mitochondria and the electron transport chain in the membrane of the mitochondria. So NADH and FADH are being produced by the Krebs cycle, are easily utilized in the electron transport chain for the production of ATP. About 2.5 ATPs are produced by 1 NADH and 1.5 ATPs by 1 molecule of FADH, like we have discussed in the summary. 3 NADH are produced through one down of Krebs cycle and 1 FADH. So 3 NADH and 1 FADH, 9 ATPs in total in electron transport chain, plus we also have 1 ATP that is directly produced in the Krebs cycle. Actually, GTP is produced in the Krebs cycle here, and this GTP is further utilized to convert an ADP to ATP directly. 1-acetyl-CoA generates approximately 10 molecules of ATP through one down of Krebs cycle. Since previously in glycolysis, one glucose was split into 2 pyruvate, which further converted into 2-acetyl-CoA molecules, which entered in the Krebs cycle. So 20 ATPs are produced per molecule of glucose in the Krebs cycle.
And that is all. You are done listening to the group presentation of Group 5. Remember that Krebs cycle is a series of reactions that produce molecules that are important to our body. It is the main source of energy for cells and an important part of cellular respiration. There are 8 steps in the cycle, and the citric acid cycle oxidizes 2 carbon units, producing 2 molecules of carbon dioxide, 1 molecule of GTP, and high energy electrons in the form of NADH and FADH2. That is all. Thank you for listening and staying until the end. Goodbye.